So I'm going to give the mic to Dr. Jalal Taneera.
Thank you very much. Thank you for the presentation, Hamda. I'm going to share my slide now. Okay. Okay. Yep. So first of all, thanks for the invitation. I'm really glad to be with you and talking about my work. My name, of course, Jalal Taneera, and I am assistant professor at the College of Medicine, Sharjah University. And my talk will be about the identification of novel genes that involve in insulin secretion and type 2 diabetes. And I will take GPR 183 gene as an example for this talk. So to start up, just a few words about type 2 diabetes. We can say it's a complex and multifactorial disease that ascribe for an interaction between common genetic variants and environmental factors such as obesity. The type 2 account for more than 90% of all diabetes cases worldwide. So far, Genetic Wild Association study have identified more than 140 genetic variants that associated with the disease. However, these 140 explain only probably between 10 to 15% of the heritability of the disease. And in most cases, these genetic variants, it's not really clear how they influence the eyelids function or how they involve in the pathophysiology of the disease. So based on that, this has really provoked scientists all over the world to develop a new strategies to identify putative or novel genes for type 2 diabetes. However, this has been all the time difficult because I would say 10 years ago, we did not have an access for a human pancreatic iris, which is really a big obstacle when it comes to diabetes research. So we were fortunate, I would say, 10 years or 9 years ago, when I was working in Sweden at that time, to start building up a collaboration between Lund University and something we call it the Nordic Network for Iris Transplantation, where they start to provide us with the human pancreatic iris. We were able to build up really a good, one of the biggest cohort when it comes to the human pancreatic islets. So at that time, we were able to collect almost like 200 islets from 200 donors. Sorry, we collected two islets from 200 donors. And these islets, of course, must be really characterized before doing anything. For example, we take, for example, some of these biomedical information about age, BMI, hemoglobin A1C, gender, insulin secretion, and etc. As you can see, this is on your left hand or on your right hand, I don't know. You can see this is one of the human pancreas that we used to get from, of course, from dead cadaver donor. And we start to isolate, which is a very complicated process, to isolate the human islets that you can see it here. So these human islets that are responsible for the disease represent all, by max 1 to 2% of the total volume of the pancreas. And here you can see a kind of immune staining for all the type of the cells in the human pancreatic iris, where beta cells represent 70%, alpha cell represent 20, delta 10, pp5, and green cells present 2%. So these islets, we used to take them and subject them to RNA sequencing or a microarray. We started first at that time with the microarray, and then we moved to the RNA sequencing. And we were able to combine data from all, from the human islets gene expression, in addition to the genetics and function, to build up a global map for the genes that associated with the islets dysfunction and type 2 diabetes. The roadmap that we used to do at that time, of course, is to compare the gene expression between diabetic islets and non-diabetic islets. Doing this correlation, we end up with some genes, of course, many genes that could be differentially expressed by doing such analysis. Of course, you need to end up with a sort of ranking. I mean, what is the important and what is the least important genes that you need to follow up and to validate. So to rank the gene and give them a priority, we used to have 5 different criteria. For example, whether this gene has any kind of genetic variants that involve with the disease, whether this gene differentially express between diabetic and non-diabetic when it comes to beta cells, or when it comes to to the islets, and then whether this gene correlates significantly with the hemoglobin A1C or insulin secretion. So based on these criteria, we used to rank the genes from 1 to 100, and of course, the less rank, the better chance that we need to validate this gene. So I would say doing this and having a list of these genes, we comes to the functional validation that we use, of course, to do in rat NS1 cell, which is one of the best model for translation research to diabetes in our community. What we used to do is to silence the target genes by sRNA and then try to follow how the silencing of this gene affect the beta cell function when it comes to when it comes to the insulin secretion, for example, when it comes to the insulin production, when it comes to a key pancreatic genes that involve in insulin secretion. So this is like the main road that we used to do. Doing this, we were able to publish several papers about novel genes for type 2 diabetes. And we're still in process to publish more and more using this approach. So this is the first part, but I'm going to talk about another specific talk today. It's about the G protein coupled receptor family. As you know, this is the largest family of integral membrane protein involved in microbiological process and pathologies. Remember that mostly 50% of all modern drugs and 25 of the top 200 best-selling drugs are estimated to target the G protein coupled receptor. They transduce, of course, a signal mediated by diverse signaling molecules such as ion, peptide, lipids, photon to induce different intracellular function. And they bind, of course, the ligand to activate different G protein. If you look at the literature, there are more than 700 G protein coupled receptors that have been identified. However, 100 of these G protein coupled receptors, they lack defined endogenous ligand. Thereby, we call them orphan receptor. These orphan receptor, their role, expression, implication, insulin secretion, or in human pancreatic islets are poorly understood, as well as a study on uncharacterized orphan receptor are of great importance to expand the therapeutic target for diabetes. If you look at the table here, you will see 4 different drugs that have been, or they are under development now, that based on the G protein coupled receptor. We have G protein 40, GPR 190, GLP receptor and glucagon receptor, GLP receptor, and GIP receptor. So these drug under development for type 2 diabetes. So we thought maybe we can expand this list by looking for another target using these 100 orphan receptor from human pancreatic islets. So what we have done here, we used the International Union of Basic and Clinical Pharmacology database to list all the orphan receptors. We end up with 91 orphan receptors. So thereby we use the microarray expression and look at their expression profile in human pancreatic islets. By doing this, we end up with four different categories of orphan receptors. Some of them, they have low or none or no expressed in human pancreatic islet. Some of them, like 21, they have a low expression. 35, they have a moderate expression, and 5 of them, they have high expression. When we correlate, if you look at the right figure here, when we correlate the expression from the microarray with the RNA sequence, blotting them together, we got really nice positive correlation between the two different technology. So this is the first step. The second step as well, we even characterize these orphan receptor in the insulin cells, which is, as I mentioned before, it's the translation model that mostly we use when it comes to diabetes research. So this shows us that there are like 55 G protein coupled receptor common between ants and the human islets. Remember that the ants one is rat. So there are 55 common. There are 36 that mostly you will find it in the human, and there is 0 G protein coupled receptor that you can find it in the insulin cells, but not in the human islets. So what we have done next is to do a differential expression with all these 91 receptor in diabetic and non-diabetic. So we have done the test in two different ways. First of all, we compare diabetic islets with non-diabetic islets. Or, of course, when I say diabetic, this is the diagnosis that the physician has made and sent it to us. But in the lab, we have done our own category here. We use hyperglycemic versus normal glycemic. So this is the category that we have done in the lab. So doing these two different analyses, we end up with several genes that differentially express. Among them, we have found the GPR 183 and GPR 75. They are a kind of overlap between the two analyses. So we thought that this is something very interesting, that it shows us that these two genes are robust in terms of the finding. So doing more research on these two, GPR 183 or 75, unfortunately we found that GPR 75 had been implicated previously with insulin secretion, but not the 183. Thereby we focus on our research on the 183 only. When it comes to the 183, you can see it here in the microarray that it's expressed there. Of course, the expression is not that high. It shows that it's in the lower percentile in the RNA-seq. However, doing immunostaining, we were able to show that the GPR 183 is expressed in the human islets and mainly co-localized with insulin secreted cells, but not, for example, the alpha cells. As well, we look at the GPR 183 in different metabolic tissue like fat, pancreatic islets, liver, and muscle. And in all of these tissue, GPR was expressed there. So that was the important startup. So what is GPR 183? It's not that much known about it, but we can say a few words about it, that it's Epstein by virus-induced gene 2, that it's highly expressed in the spleen. It's required for human immune response, including B cell migration and T cell dependent antibody production. As well, it has been shown that there are metabolites called 7-alpha-25 dihydroxycholesterol work as a potent agonist for this GPR 183. So we start up first to look the role of GPR 183 in insulin cells by knocking down this gene and look whether knocking down GPR 183 will influence the insulin secretion in the cell line. So we If you look at the figure A here, you will see the silencing efficiency after silencing these two genes. We have used GPR 183 and 75. We have used 75 here as a control since it has been indicated. So we thought, okay, let's keep it on and use it as a control. So the efficiency of the silencing, as you can see, almost 80% of the genes on these NS1 cells. And when you look at the insulin secretion here, you will find that knocking down GPR 183 in NS1 resulted in a significant reduction of the insulin secretion compared to the negative control. So next, we examine whether the silencing will influence the insulin production. Unfortunately, although we find a sort of reduction, but it was not significant. More we look at the consequences of the GPR silencing on beta cell function genes such as insulin, of course, gene, PDX, and MAF. These are very important regulator for beta cell function. So we, as you can appreciate from the figure, that all of them should to be influenced by significantly down regulated. So we have a finding here. We thought, okay, since the 7-alpha-25 dihydroxycholesterol has been proposed as agonist, so why not to try? So fortunately, as you can see here from figure A and B, when we use different dose from the agonist 7-alpha-25, 0, 10, and 100, 1000, we found that there is a sort of insulin potentiation for insulin secretion in human pancreatic islets in a dose-dependent manner. So the more you give 7-alpha, the more the human islets will secrete insulin. As well, we found that these agonists as well protect the cells by increasing cell viability if we culture these cells in high glucose medium. Knowing that high glucose medium, if you culture your cells, that will create a sort of apoptotic environment for these islets. But what we see here, that the 7-alpha protect these cells in high glucose culture medium. So we were very happy about these data. I don't want to talk about these two figures here because of the time, but maybe I can move for the next. So the next, we try to study different type of mechanism for the GPR 183 by looking or investigating whether this gene or this agonist influence cell proliferation or cell cycle in the NS1 cells. So doing thymidine incorporation here in different dose of the 7-alpha dihydroxycholesterol, we found that a significant cell proliferation in a dose-dependent manner, as well as we doing a cell cycle analysis for the NS1 cells culture and 7-alpha-25 dihydroxycholesterol, we found a significant increase in the S phase and the G2M phase, but not on the G0 or G1 cell, which is in a way fit to the cell proliferation data. Lastly, we look at the effect of the 7-alpha-25 dihydroxycholesterol on certain beta cell function gene, such as, of course, insulin gene, PDX, GLUT2, insulin receptor, and glucose kinase. So these are, again, a very important genes in the beta cell and when it comes to its function. So what we found that the 7-alpha influenced the insulin gene, it influenced PDX, it influenced the insulin receptor, but not GCK or GLUT2. So by this, we conclude in our data that this study shows a comprehensive profile for all the orphan G protein covered receptor expression in human pancreatic islets and in NS1 cells. As well, we identified the GPR183 is a novel gene for type 2 diabetes. We showed that 7-alpha 25 dihydroxycholesterol potentiate insulin secretion, reduce glucose toxicity, and increase cell proliferation. As well, important thing here that we show for the first time that show a novel mechanism for cholesterol metabolized in insulin secretion. And this is very important because we were the first to show that cholesterol, because it has been thought that cholesterol is a bad boy, but here we show no, it has some good effect. But this we can say finally that we believe that GPR 183 could be an attractive drug therapy for pancreatic beta cell function. And lastly, of course, I need to acknowledge all the people that share with me the work in this study. Of course, Al Jalila Foundation for their support. Of course, our colleagues at Lund University, Sweden, for their support and for data that we have got from them. In Sharjah, of course, this is my lab here. So this is our co-worker that shared the work with me. I would like to acknowledge all the people in the Diabetes and Metabolic Research Group and the Iron Biology Research Group. Finally, of course, this work had been supported by a grant, of course, from Sharjah University. And thank you very much. I hope I'm on time.
Thank you very much, Dr. Jalal. It was a very interesting presentation. Now, Dr. Ihsan will present.
Sharing.
Dr. Jalal, if you can please stop sharing. Okay,
Yes.
thank you.